Hi all, this lecture is about an overview of drug discovery and development. We shall start the journey of drug discovery with a little note on history of insulin discovery, and we'll be discussing about various stages of drug discovery, preclinical studies and then clinical trials. We'll be also seeing about the regulatory guidelines pertaining to drug discovery and medical ethics. The first step in the pathway towards discovery of insulin was the target identification and validation. The scientists they removed the pancreas from a dog and found out that the dog died of diabetes producing ketone bodies. So they knew that pancreas was the site of pathogenesis of diabetes, and they validated it by removing the pancreas and also by studying that a chemical called insulin was missing from the pancreas, which results in the diabetes. So this is the step where the target is identified and validated. Later in 1921, Frederick Banting, who was a surgeon, and his assistant Charles Best removed insulin from a dog's pancreas. They prepared an extract out of it and injected into another dog with severe diabetes and kept it alive for 70 days till the extract remained. This step we call it as lead identification and optimization in drug discovery process. So here by lead we mean insulin. So they identified the lead, which is insulin, and optimized it. The insulin extract, which was prepared by Banting and Best, was crude, and hence they wanted to prepare a more pure form of insulin, and hence they collaborated with J.P. Collip and John McLeod and developed a refined form of insulin from the pancreas of cattle. And this final formulation of insulin in a refined manner was injected to the first patient, a 14-year-old boy, Leonard Thompson, in Toronto Hospital in January 1922. So he became the first person to receive an refined product of insulin. So this is the story of insulin discovery. The drug discovery and development process at present is rather similar to the discovery process which was happening in the past, but in a more technologically advanced, structured, and strictly regulated manner. These are the stages of drug discovery and development process. So stage 1 is the drug discovery process, stage 2 preclinical development, stage 3 clinical development where there are different phases of clinical trials, and finally regulatory approval and marketing of the new drug. So you can see numerous molecules will be screened in drug discovery process, out of which some will be developed in preclinical phase, and very few will go for clinical trials, and finally you will get one drug after the regulatory approval. So the whole process is time consuming. It takes many years for a new compound to come to the market. The entire process of drug discovery and development is time consuming. It takes almost 15 to 20 years for development of a new drug molecule and market it. And it is very expensive. So it requires millions and millions of dollars, and it needs to meet extremely stringent regulatory requirements and environmental concerns, and most of the patent period will be spent in development process, and once the patent expires the profit will be drastically reduced. So these are some limitations and hurdles the process of drug discovery and development has to undergo. We can divide the process into discovery part and development part. So in discovery part we have target identification and validation, then screening of libraries of compounds and selection of hits, and development of hit to lead and optimization of the leads, and finally a new chemical entity discovery or identification of a potential drug candidate. So till this stage we call it as discovery phase, and then once the new drug candidate is identified, then it enters into development phase, which involves preclinical and clinical trials. We shall see different stages in the discovery process one by one. So the first step is target identification. This means identifying the function of a possible therapeutic target. So this can be a gene, a protein, ion channel, or any target which is involved in pathology of the disease. And target validation. So this means you have to confirm that the molecular target is directly involved in the disease process, and also when you modulate or modify the target, it should be likely to have a therapeutic effect. So this is target validation. While validating the target, we should make sure that the target is druggable. So that means the function of the target should be altered in the desired fashion by an organic molecule. So the molecule which we are screening should be able to bind with the target easily and modify the function. So that is druggability of the target. Next step is identification of hits. Hits are molecules which bind to the target and modify its function. So how we identify these hits? We screen compound libraries which contains thousands of molecules, and by screening these molecules we identify those molecules which can bind to the target and modify its function, and we call them as hits. So the sources of these molecules in the libraries can be from natural sources like plants, animals, microorganisms or minerals, or it can be synthetic. The final step in the drug discovery is lead generation and optimization of the lead. All the hits which we have identified cannot become a potential drug candidate. So we will have to determine various physical, biological and chemical characters of the hits, and then identify the most likely molecules which can become leads with improved potency and reduced off-target activities. So once these leads are generated, we will improve the target specificity, selectivity and potency of the leads, and finally the leads will be optimized, and these optimized molecules or leads will subsequently be developed as a drug. After the discovery of potential drug candidates, they enter into development phase, which involves preclinical and clinical phases. So preclinical studies are performed in vitro, in vivo, ex vivo and in silico models to obtain basic information about the safety and biological efficacy of a drug candidate before it is tested in the final target population, that is humans. The preclinical development phase cannot be designed blindfoldedly. We should have a framework to ensure that the preclinical development phase supports the intended clinical trial design and therapeutic use. So this framework we call it as target product profile. For example, if you are going to develop a drug for Alzheimer's disease, the drug should be able to work in elderly patients. The drug should be able to cross the blood-brain barrier to reach the target site of action, and the drug formulation should be designed in such a way that it is easily taken by the patients. The bioavailability is more if you are going to give in a tablet form, and since the patients will be in extremes of age, the drug should be safe and free of any toxicity to liver and kidney, and there should not be any contraindications. So these are the factors which we have to keep in mind, or these factors should be predefined so that the intended purpose is served. Different studies are undertaken in preclinical development phase. We have efficacy studies to confirm and validate the therapeutic effect of the drug molecule, and bioavailability studies, pharmacokinetic studies to study the Cmax and Tmax of the drug. We have dose range finding and toxicity studies to determine the maximum tolerated dose and to identify any observable sign of toxicity. Animal toxicity studies form an essential integral part of preclinical phase. We have different types of toxicity test. Acute toxicity test use two species of animals and the drug is given through two routes. Here we determine the no effect dose, maximum tolerated dose and LD50. Then we have subacute or chronic or subchronic toxicity test. Here three doses of drug is used in two species. The test is conducted for 2 weeks to 3 months. And determination of biochemical and physiological effects of the drugs in different organs of the animals is studied here. Chronic toxicity test involves rodent and use of one non-rodent species. And this test is conducted for more than 6 months. The chronic toxicity test is especially needed if the drug is going to be used for a prolonged period in humans. Here, the same effects which we see in the subacute toxicity test is confirmed. We also have toxicity test to study the effect of the drug on reproductive performance, the carcinogenic potential of the drug, and also mutagenic potential of the drug. So these are different types of animal toxicity tests. At the end of preclinical phase, the active pharmaceutical ingredient will be optimized and the final drug product will be formulated by adding necessary excipients and stabilizers to the API. At the end of preclinical phase, investigational new drug application will be made to the regulatory authorities. So only if the IND application is processed and approved, the drug will be entering into the next stage of development process, that is clinical trial phase. You should be familiar with certain regulatory guidelines which should be followed during drug discovery and development process, especially preclinical development phase. GMP, good manufacturing practices, or set of guidelines which should be followed during manufacturing and formulating the drug. GLP, good lab practices, good laboratory practices or set of guidelines to be followed while conducting animal experiments. CPCSCA, Committee for the Purpose of Control and Supervision of Experiments on Animals, is the regulatory authority which gives institutional approval for conducting animal experiments. IAEC is Institutional Animal Ethics Committee which functions in every institution, and this approves the protocols for animal studies. CDSCO, Central Drug Standard Control Organization. This comes under Ministry of Health and Family Welfare in India, and this is the counterpart of FDA in US. So this deals with all the drug approval and regulation. So this is the ultimate drug approval authority in India. And Drug Controller General of India is the drug approving authority which functions under CDSCO. These are the references. So we shall see about the next and final step in the drug development process, that is clinical trials, in the next lecture. Thank you.